is to support faculty, staff, and of course students, mainly, I would say, across university in the form of the adequate access provision and services. This is actually our key function. So the next, it's conducting experimental analysis for you guys, mainly for students again. Why? Because usually the faculty and staff, they know really what they kind of want to do. They know really the instruments mainly, but the students, we are kind of teaching them, we're guiding them through our instrument park and so on. So maximizing the efficiency of research equipment utilization to increase access, it's also our key. Why? Because our goal is the less time you are spending here, but with the most outcome, it's better for us. Of course, why? Because in that case, we can cover more students. The last kind of part, equipment maintenance and servicing, actually is not related actually to you, but it's, of course, it's our also main goal. We are located on the 6th floor of the block C4. And myself, Aidos Baumuratov here, and my colleague Aigul Kusanova, she is a research technologist. We are two of us, we are actually maintaining this laboratory. My presentation, actually, I would start from this. My presentation, we are currently, and as a core lab, we are not doing research, academical kind of research, like you heard before the speakers, like different professors. We are helping the researchers, including professors, including staff members, including arrays, including, of course, students of bachelor's or master degree or PhD students, to conduct research. So our goal, again, so you come to us, if you don't know how to use that or another instrument or any instrument that you need to use, we will teach you and guide to you. And especially we will consult to you, which is actually more important, because sometimes PIs, professors, they don't have time, maybe enough time to explain to you some kind of details of the experiment and so on and so on. And in that case, we actually here, we are really well trained and experienced. And actually in the past, I was kind of group leader and doing, of course, also academical research. And currently I'm lab coordinator of this core facility. So my kind of presentation is not really about doing research. My presentation about what we have, what we can do, and what we can provide to you. So first I want to start with this microscope, which is called confocal laser scanning microscope. Here you can, see I will not tell you all these technical details. Actually, this is not really matters. What is important, what you can do with this microscope. So what you can do is written, small part of it. For example, if you are a student of life science, so for example, protein localization, cell characterization, morphology studies, I think these kind of words, it's not kind of unknown for you. So you should be familiar with these assays, of life science research. If you are, for example, student of School of Engineering, or electrical engineering, or another kind of engineering, or from the material science. So for example, for you, probably will be interest of like a surface analysis, topology studies, or depth result spectroscopy. So all these assays and the techniques, and actually I wrote here, not all of them, because actually the list of the assays is actually pretty long. It cannot be I cannot put everything in this presentation. And with this microscope, we can do everything. So for example, starting, if you are interested in the saying by kind of simple words, if you want to localize protein in your live specimen, we can do here. If you want to do the topology of your material science, you did some new material and you want to see how your structure of your new material looks like, we can do that easily without doing any sample preparation. Compare, for example, to electron microscope that we have also, I mean, downstairs with another, my colleague, he will be presenting, I think, in a couple of weeks ahead. The next microscope, it's called fluorescent inverted microscope, is designed. Also, all these technical details, I think you could find easily on website. You can later like, again watch it on YouTube and see all these technical details. This is actually not really matters. What matters is this system is designed to do live cell experiments, and not on this microscope you cannot do studies working with a kind of non-live specimens, let's say. So if you are working in life science unit or in biology studies, or for example, maybe you are a material scientist who is collaborating with the life scientists, and then you did some kind of a new material and you want to see that material's biocompatibility. Then of course we can go here and do some research, for example, like biocompatibility, testing different kind of, by using different kind of assays, we can see, I mean, your material is good for using together with cells or not so. So here is also some list of assays that can be performed on this microscope. So it's live cell imaging, protein localization, again, cell characterization, kinetic studies, so it's a prolonged cell recording, wound healing, like a very useful and popular actually assay, and morphology studies. So unique thing of this, it's this incubation chamber. Inside of this incubation chamber, we can make it that way that inside of that incubation chamber, your cells, or let's say small organs or small animals, like zebrafish or Drosophila larvae, that they can feel happy. Why? Because they usually live, I mean, at 36 degrees and with the humidity and so on. So inside of this incubation chamber, we can reproduce that condition. Another microscope that we have, it's called spinning disc confocal microscope. So actually this is very interesting device. It's actually very new. We received just last year, in like almost 1 year ago. This microscope combines strong kind of sides of both previous microscopes. It's also designed only and mainly for the live specimens. So for the live cell imaging, this, I mean, if you want to do the live cell experiments, so you need to book this instrument. And also the list of assays, you can see here what we can kind of provide to you, we can train to you, we can consult to you, and so on, and make kind of expertise analysis and experts kind of conclusions for you. But of course, the sample preparation, it's on you. Before that, of course, you can come to us and we will saying that it's better to do this way or it's better to do that way. Before doing so, again, you can go to your, of course, it's better to go to your PI and ask, understanding the fundamental and functional goal of your kind of study. Also some technical details, it not really matters. What matters, I mean, this unique device, it's actually, I would say it's really unique. And in Kazakhstan, all these 3 microscopes shown, I mean, before by me, they're really unique, and I would say in 1 or 2 pieces only in the whole Republic. So our students are really kind of lucky that we have this equipment, and this equipment is maintained very well. And I mean, our university hired, I mean, us, experienced and qualified staff to do this all and help actually our researchers in which, I mean, also the students, our main customers or users are students, mainly master students and PhD students. And unfortunately, or maybe will be future, this number will be growing, and it's only a small number of bachelor students. This piece of equipment, it's called kind of imaging workstation. Why it's important, I will show you later some samples, and you will probably understand that. I also will describe it. This is just a normal PC, but it's not really a normal PC. It's a really powerful PC on which we can analyze our data. Why? Because imaging, it's actually a word that concludes of acquiring image and analyzing image. So acquiring image is done using that microscopes. Analyzing of images, especially of 3D stacks or 4D stacks, it means 3D plus time, especially if you are doing prolonged studies like hours and sometimes days, continuously you're acquiring images. Imagine this is a gigabytes of data. And for that reason, we bought this really powerful computer with some of these technical parameters you can see, and installed the list of this software, which is really from world-leading companies. And of course, also we can give you some guidance how to use it, how to analyze the data. But before doing so, again, most important, you need to understand what you want to get from your results, or at least you need to know the glimpse of, I mean, what are you doing? So here is a, I don't know, this actually, so here is a list of, I mean, most commonly used analysis done on this workstation. So it's quantification of image, deconvolution, morphology, cell content characterization, or cellular dynamics, and so on and so on. There are, I don't know, hundreds of different assays what you can do. Now, okay, now I have small problem already. No. Now I will show you, because showing this all microscopes is kind of fine. You are thinking, okay, I mean, maybe it's a little bit boring, but what we can do really, because I know you students, I mean, many of you guys was here, I've been showing them, I mean, my lab and so on, but you're asking what we can do because showing one image is worth of 1,000 words. So now I will show you some examples what we could do, but it's only part of what we could do. And based on this, you can actually realize, and at least you will remember better when I will be showing this. So for example, I start with this. This is a reconstructed, first it was acquired on the confocal microscope on the upper left side. 3D image of plant section. So we cut the plant, we stained with the fluorescent dyes, and then we acquired by laser dissecting in 3D. And then these 3D slices we reconstructed. And this is actually representation in the special software, how it looks like. So this, for example, this is a cell walls in the plant, and so on and so on. The next one, this is a merge of three images, two fluorescent image, DAPI. DAPI stains the nuclei. In the nuclei, DAPI actually stains DNA. DNA located mainly, of course, in the nucleus of the cell. And here you can see the cell culture of, it's called sushi cell, we call it like this, but it's SHS55Y cells. And the green one, it's a mitochondrial network. And the gray background, it's a transmitted light. So the next one, this is a high magnification, high resolution image of single cell. It's a HeLa cell stained for the nucleus, for the actin. It's a protein of the cytoskeleton. And the red, it's a mitochondria again. So for example, if you're interested for example, number of mitochondria per cell. So what we could do, we just take a single cell image, and by using the special software, we quantify this number of mitochondria. For example, if this is a kind of topic of your study or something related to this. And if you want to do more, then we can do 3D section, reconstruct it like we did in this sample, and do it in 3D. So it means we will be collecting stack of images, reconstruct it, and quantify it not in 2D like XY, but XY and Z direction. This is actually more precise, and this is much, much better because the quantitative data is much more precise. So this on left downside, it's for example, colon biopsy study. This image was kind of done using the 10x objective, and 144 images was stitched together in order to find out the neuronal network in the gut, because some part of the neurons in the human body are located in the human gut. So for example, this, I mean, based on these samples, we can at least have the idea what we are doing or what the research are more or less what, I mean, really, this is a live research. This is not like theoretical things, like this is what we have been doing and what we can do. Next, also still, again, samples, I will show you. So when I was saying about deconvolution, if you remember, deconvolution, it means on this sample in the middle, deconvolution, this is non-deconvolved image, this is deconvolved image. So as you can see already, the conclusion is that through deconvolution, we are finding the light which was scattered and lost. This is a simple explanation. So we are applying some equations using specific software, and that software allows us to find light that was scattered. As you can see, it is a blurry kind of image. And after applying some equations that was used by different software, we are finding the light that was scattered in voxel in 3D, I mean, direction in X, Y, and Z, and we're reconstructing image back. So this is the original image, and this is a deconvolved image. So this is also possible in our facility. For example, how it can be applied practically. You did kind of sample preparation on your, I mean, cells or small fish or small Drosophila larvae or something. But you did sample preparation not really good. Still, it is possible to find something and to actually to get data from that your sample and kind of recover it. So kind of lost data, it's not completely actually lost. It's lost in the by diffraction of the light. So that data can be reconstructed back, and we can kind of find it back. On the left side, upper, on the first I will describe, this is a, you see, this is a small zebrafish. This is a larvae, kind of, I think it's a 2 days old larvae of the zebrafish. This is 10x objective, 28 images. And on the upper part, so this scale bar, it's a 200 micron. And this, if you see this, I'm not sure, do you see it or not? This blinking kind of things. This is the blood cells in the vein of the tail of this fish. So this green, what you see here, it's actually genetically modified protein, GFP, that was introduced into the blood cells of this zebrafish. And then by doing the live cell imaging, we were recording this, I mean, alive. So we have put under the microscope this fish, and doing this live cell imaging. So the next on the right side, now I come to the to the kind of sample that probably would be interest of the material scientists or students of the engineering department or kind of non-life science students, I would say. So this is, you can see that we did some collaborative work with a professor from School of Engineering, and that we were researching the topology of perovskite solar cell surface. So they did some new development of the new solar cells, perovskite based. And then they asked us to do the topology study. So the topology, we just use, if you see it here, we are giving them the analysis of the surface by like surface roughness or surface, mainly surface roughness. And if they're using the specific ion kind of pump, bombarding these perovskite cells to find out where is the layer in which, I mean, from which these photons get excited. And then using by confocal microscope, I am kind of cutting in 3D this material and finding on which thickness the most, I mean, on which thickness that layer lays, kind of. So for example, from the surface after 10 micron, there is a kind of layer in which the photons gets most excited. So this kind of analysis we can also to do. So this was the samples. Now I will tell you about our kind of smaller part, but it's also very important. We have a flow cytometer. So we have 3 microscopes and 1 flow cytometer. It's called Attune NXT. It has 3 laser lines. We can collect up to 13 parameters. So it's 11 fluorescent and plus physical parameters. Physical parameters, it's cell roughness and cell size. These 2 physical parameters, cell roughness and size, tells us different information like a cell, what kind of cell it is. Because, for example, blood cells, they are smaller than, for example, epithelial cells. Epithelial cells more kind of rough than the endothelial cells, and so on and so on. So this flow cytometer is designed also for the live specimen. This is a short picture, I mean, theory behind it. So in cells in suspension, we are putting, and by laser excitation, and when before that we are actually binding to the surface of the cell antibody, and to the antibody we're actually binding the fluorochrome. And this fluorochrome under the laser light gets excited, and we are registering light back. And here is a list of assays that we can do. So the kinetic assays, again, microparticle analysis, apoptosis, DNA content analysis, cell cycle, cell proliferation. How the data looks like when the kind of results after experiment kind of done, it's like this. So for example, in this experiment, the whole blood of cell was loaded. And then by doing flow cytometer experiment, we can separate each kind of blood cell into separate groups. So for example, here, this is a granulocytes, this is a monocytes, this is the lymphocytes of the cell. And this part, it's actually the trash or dead cell. So this is the most easiest experiment that can be done on the flow cytometer. So here is a list of the projects. Just I have chosen to show you that we're actually involved in. Because I said before, we are not doing research ourselves. We are just collaborating with the different PIs from the different schools, from the different departments, and helping them to do the research. So here is the list. I will not just read it for you. You kind of read it yourself, and you can see that we can cover different kind of broad aspects of different research, from life science to the kind of material science research. Now, how you can access all this equipment and how you can kind of contact us and find us. So first location, again, I will repeat, we are located on C4, 6th floor. And if you want to book an instrument, then you need to go to QReserve. So you open this webpage, myqreserve.com. And then using your NUEDU account email, you create your account in this system. Then after you enter, you finding Nazarbayev University and then clicking core facilities. After doing so, you will see this page and the list of equipment that is available in our core facilities. So you're clicking it, and then there will be kind of a reservation calendar like here. You just click on any day that is convenient for you, of course, before contacting us. And then by approving, you just come to us and we will be doing some research or experiments for you. But of course, before, it's most important, you should contact us by finding, easy, by writing by email or by phone call or whatever you like. It's better actually by personal, because it's easier to describe your project and we can more easily help to you. That's it from my side, because this is it. That's all what I want to say to you. Do you have any questions or comments? You are welcome.
Can anyone from outside of Nazarbayev University book laboratory for their purposes?
Yes. But in that case, it will be a little bit different. It depends from which institution are you from. It really depends. In every case, it could be different. It depends again. If it is a scientific institution like a National Center of Biotechnology, it's different that if it is like a commercial company. For example, some commercial company can bring us some polymer to see the biocompatibility. But this is really pure commercial product and they want to see kind of really analysis. In that case, it's analysis and it will be different and we will charge them. If it is, for example, National Center of Biotechnology, it depends on collaboration. If we are collaborating with them, it could be free. Yeah, if we are not collaborating, we will also charge them, but in that case, the price could be the difference. So in every case, we should be you should be contacting us and talking to us. But of course, for the students, for the staff, for the research kind of community in Nazarbayev University, all these equipments are free. Another question?
You said that mostly master and PhD students are involved in the lab. Are there any, at least one undergraduate student who is using your equipment?
Yes. Now we are starting. I would say this because I am really involved in this. Now I'm a co-PI of one project where is one undergrad student is involved. She is from School of Science and Humanities, and the project I can only just shortly tell it because we're just starting. It's creating organ on a chip. So it will be liver on a chip. So it's artificial liver. And she is undergrad, yes. Why? I can tell you why mainly master and PhD students. It's because I think the undergrads just started and they need some time just to get in and to kind of to understand what really is in and what professors they really like, what research they like. And when they're really at third or fourth years, they already kind of know what they want, at least in the scientific world. And then they start to really come to us. Any questions?
Yes, I have a question. And how much time does it take to take a permission?
To take a permission?
Yes, to use the lab.
Depending on the instrument. If you want to do the self-use, then you need to get approved by, I will be doing for you a training. After that, you will be kind of passing my exam. And if you will pass, you will get a self-use. But if you don't want to do a self-use kind of experiments, if you don't want to learn this kind of instrument or technique, then I can do it for you or my colleague. And in that case, it's really fast. You just bring your sample and we'll be doing experiment for you.
Okay, thank you.
How does this course work? Do you have like any semester-wise or like annual courses that are offered to students?
Yes, actually now I wrote today email to the all, and I think after this online session I will write to the students all. Now actually I will be reading the course. My course is twice per year. It's very short. It's called Introduction to Light Microscopy, and it is only 3 days, about 1 hour every day. But this is not how, it's not about to get access. It's about to get, to give you some introduction about like how is light is passing through material, how is optical elements are constructed in microscope and so on and so on. So it will be better for those of you, and it will be of interest of you who want to do some microscopy things, who wants to do some like a flow cytometer experiments, who wants to do the research related to the kind of using the microscopes, different kind, optical microscopes, confocal microscopes, fluorescent microscopes, and so on. I just sent email today. Now I will send this email about registering to the course to the students. So you can register and participate if you like. It's also via Zoom, because now everything is via Zoom.
Do students get some kind of a certificate or credits for participating?
No, this course are voluntarily, and there is no any credits on it, no.
So is there any like certificate to show to someone else or to some company that a student learned that?
Unfortunately not. We are not certified to do so actually, even because we are a public organization, we're a university, we cannot even do that.
But after this course, students will be allowed to for self-use.
No, this after kind of listening or participating into this course, you are not granted by access. Access, it's a personal. You're just contacting me. We are scheduling the days when you will be kind of trained. It depends also on the instrument. For example, on confocal microscope training, it's 4 days, from 1 to 2 hours, 4 days consecutive. After that, you are kind of passing my exam, and you've passed it. I am giving you the permission to use that instrument at your own. But this course is just to introduce you to this theory of light, theory of fixative to prepare a good sample, theory of like microscopes, and so on and so on.
Okay, I got it. Thank you.
You're welcome.
Hello, Aidos. This is Irgali. I'm Senior Lab Coordinator, School of Medicine. I have a couple of questions. So which of these confocal microscopes does any one of them have on-stage incubator with CO2 supply?
With CO2 supply, no. But if you really want to do, I would recommend to use, to use non-CO2 independent medium.
Okay. So
it's better actually. But if you really want to CO2 gas, we can actually do that. Of course, it will take some time from us to kind of prepare all this, but we can do in some case if you're kind of, for example, your research cannot be done not using this kind of CO2 gas. But in other cases, we recommend to use CO2 independent medium.
So, for example, if a School of the Medicine faculty or researcher want to use your facility, can they can they have like access to CO2 incubator and biosafety hoods? Because bringing the cells from afar.
Yes, this is a very good question, Yergali. I know this, and like actually the School of Medicine researchers and students, this is not first time you're asking. Yes, because many of you, students and researchers from the School of Medicine asked this, and I did this, and personally, actually, I did most of it by myself. We, of course, together with my colleague, Aygul, we just put it inside, I can show just actually. So we put it this hood, bio hood, and we put a CO2 incubator, and of course the fridge. So now you can prepare you can bring your sample here, and in our lab you can prepare your sample just before doing some microscopy studies, microscopy kind of experiments. Now it's available, yes. Answer is yes.
Yeah, thank you. That's it for me.
Okay, I guess there are no more questions for you. Thank you very much for this presentation. It was very interesting. We also recorded the session. Anyone who missed it can make some notes.
Okay.
It was really cool. Thank you for your time. And thank you everyone for coming and listening to this session.
Yes, thank you all for attention and for time also.